So thanks everybody for coming. I'm Bronwyn Bleakley, and I'm a professor of biology here at Stonehill College. And I'm going to talk about a long-running research project, in particular the updates that came from my sabbatical, which occurred last year. But I want to walk you through my research program, which investigates how cooperative behavior emerges in Trinidadian guppies. So I'm an evolutionary biologist by training, and I'm always going to take an evolutionary framework. So evolution by natural selection happens whenever there is genetic variation for a trait. For example, here you can see that these two little mice vary in their coat color, and the two blue-footed boobies on the other side of the screen vary in both how blue their feet are, but also how energetically they dance with one another as they are engaged in courtship. And those traits experience selection depending on how well suited they are to the environment, for example. So you can imagine that this little white mouse is way better suited to this sand environment than the little brown mouse, right? It's okay, I got it. So variation in traits results from both the effects of genes and the environment. So again, we've got our coat color for mice, or we've got an energy level for our blue-footed booby dance, and variation is going to come from differences in their specific genotype and also their environment. But the environment doesn't just include things like what the temperature is or how much food is available. It also includes the social environment, and that's the environment that I am personally interested in. Okay, so people are therefore pretty familiar with the idea of natural selection, which is exemplified here by Darwin's finches, who vary for the shape of their beak depending on the food source that is most prevalent on the island in the Galapagos that they're found on. And people are also pretty familiar with the idea of sexual selection, where individuals choose their mates. But what is an underappreciated source of selection is called social selection, and it depends on who your social partners are. So for example, here we have these meerkats. They're a pretty famously cooperative mammal that lives in Africa, and you can imagine that this little matriarch is going to be more successful if she has a troop that is willing to go out and forage and babysit, compared to one who has a family that is maybe less invested in their success, right? So all animals interact with social partners, even ones that we think of as being solitary, like these tigers, are going to have territorial or aggressive interactions. These sage grouse hang out by themselves most of the time, but then aggregate in these big, intense groups during the breeding season. And then at the other end of the spectrum, we have these jacks who are with a large social group continuously for their entire life. Whenever we have these kinds of social interactions, we can see what is called an interacting phenotype. So an interacting phenotype is any trait that depends, at least in part, on an interaction. So let me give you an example. Here we have these two crayfish, and they have met up with each other. They don't actually know anything about each other, and we can't say anything about their dominance status until we've watched them duke it out. But over the course of this interaction, one of them is going to emerge as the dominant individual, and one of them is going to emerge as the subordinate individual. But you can imagine that if this one had happened to be paired with somebody who was bigger or faster or older or stronger, that they might not have ended up in the dominant position. And so this outcome reflects the properties of both individuals. And interacting phenotypes don't just apply to aggressive interactions. Again, they can be applied to these kinds of cooperative interactions. So having these kinds of interactions changes the way that animals experience selection. So again, we have our little crayfish, and he lost the fight. And now he is both less likely to get a mate, and he's pushed out to the periphery where the territory is less protective, and he's actually now at greater risk of being eaten, again, because he lost that fight. Okay, so now we're going to get just a little bit more technical. So interacting phenotypes, again, these traits that depend on the behavior or traits of both individuals, result from a thing called indirect genetic effects. And here's how this works. So this is what's called a path diagram. This is how evolutionary geneticists diagram the effects of genes. So A is for genes. I know that's intuitive, right? And then E is for environment. And so here we have this guppy. She's got a behavior of Z affected by her genotype and her environment. And so we're going to call that a direct genetic effect. And for the rest of this talk, I'm going to use color as a proxy for the behavior and the variation in the behavior. So if you see a different color, you can imagine that that is a different intensity or a different kind of reaction in the fish. Okay, so now she has a social partner who has her own genes and her own environment. And through this pathway, because the genotype of the social partner influences the interaction that they have and changes something about the behavior of this fish, this is an indirect genetic effect. And I'm just going to tell you that the strength of this interaction gets characterized by this Greek letter psi. So if psi is 0, then this goes away. There's no interaction between partners. If psi is 1, then what this fish does is totally dependent on what her partner does. Okay. So guppies are used by lots of researchers all over the world as a model system for studying the evolution of social behavior and a whole range of evolution, including indirect genetic effects, because they are native to Trinidad and the northeastern kind of corner of South America. But in this northern mountain range in Trinidad, there are a large number of these independent river systems in which everyone has a series of waterfalls as you go kind of up into the mountains. And in every one of these river systems, there is a point where, although the guppies have gotten up above the river, up above the waterfall, their large fish predators have not gotten up above the waterfall. And so we have these repeated populations that really amount to a long-term natural evolution experiment where you have what are called low predation populations, where they just don't get eaten very often, and high predation populations where they run away from big fish all the time. Okay, question? This one is, so this one is the Marianas waterfall. This one, it's my picture, but this is a pretty famous one. And so actually guppies are not above this one, but we actually don't really understand how they disperse above waterfalls. There's actually a postdoctoral researcher working with a guy named David Resnick, who's a pretty famous guppy researcher, who's trying to figure out how they get up waterfalls. So it's a bit of a mystery, but they're really good at it. Yeah. Okay, so the last thing I should say about this is that wherever they encounter big fish, they do a cooperative anti-predator behavior called a predator inspection. And that amounts to two fish breaking away from the larger shoal. They take turns being in the closest position to this big fish. And then from that, we believe that they're able to get information that helps them to assess just how threatening these big fish are. They do this by forming long-term pair bonds, long-term affiliations, primarily between females. They exclusively cooperate with non-relatives, and they engage in a whole suite of these anti-predator behaviors. But guppies aren't just a model system because they exist in this, this natural habitat. They also provide a genetic tool because people have been breeding guppies for their pretty fins and their other kind of secondary sexual characteristics. And so you have all kinds of inbred strains that differ for their color, their behavior, their physiology, their size, like every trait that you could imagine, they vary for. And that gives us a tool. So if we want to run an experiment where we control what social partners do, then we can use an inbred strain as the social partners. Or if we want to control what our focal fish do and vary what our partners do, then we can use an inbred strain. And what this does is reduce the variation in the behavior that we see that results from differences in genotypes. Okay. So then the way that we would identify indirect genetic effects looks like this. So I'm going to give you this kind of graph because you're going to see this more than once. So here I've got two different strains of guppies, these half greens and half yellows. And then here I've got two different strains of focal fish. And if the behavior on average of my focal females differs between these two strains within a partner environment, then we're going to call that a direct genetic effect, right? What's different here is the genotype of these two groups of fish. If, on the other hand, this strain changes their behavior when they're moved to new social partners, right? And same thing here, when they're moved to new social partners, then we call that an indirect genetic effect, because what changed are the genes that the social partners have. Okay, so when I first came to Stonehill, this is what our anti-predator trials look like. We have a model predator. We drop in our three partner fish, we drop in our focal fish, and then we observe how many times they approach this predator, how close they're willing to get, and how much time they're willing to spend staring at the predator, and how much time they spend shoaling or kind of schooling with their partners. And it's a pretty salient cue because they're a lot smaller than even this model, and they treat the model just like a real fish. And so here's just a cute video. Hold on, let's see if we can get it to play. There it goes. Come on. There we go. Of them swimming out, and you can see that they're kind of paying pretty close attention to what that big fish is doing. One of them's kind of stuck there because he's not moving, right? Okay. So these are real data then. So this is set up exactly like I showed you before. We had 2 inbred strains of social partners, we had 2 inbred strains of focal fish, and we saw differences in the genotype of our focal fish. So we saw direct genetic effects. We also saw change between our social partners, so we saw indirect genetic effects, and that was true for proximity. We saw only indirect genetic effects for inspections, and we saw this interaction with the shoaling, where our blue females shoaled most when they were with half-green partners, and the snakeskin females shoaled most when they were with half-yellow partners. So then I worked with some collaborators who are actually at the University of Exeter, although they're at the Devon campus, not the Cornwall campus. And we did a big experiment in Trinidad using natural populations. So we had, let's see, 250 fish. Each one of them got tested 4 different times with a social partner. We did it for 8 populations, so 4 pairs of high predation and low predation populations. And we measured that psi, the coefficient of the interaction. And the way that we did that is by taking the partner behavior and graphing the focal behavior in relation to that partner behavior, and the slope of that line is psi, right? So again, if there was no relationship, the slope would be 0. If there's a perfect relationship, the slope is 1, right? Okay. And so what we found, now we just have like the average interaction, and what we found was that that interaction coefficient varied with sex, with population, with the level of predation that they experience, and the river of origin. So basically all of those things really mattered. There was a ton of variation. One of the things that we noticed, though, was that it wasn't just that it varied. You could get an opposite reaction to that same partner's behavior. So what I mean by that is that while those coefficient of the interaction was usually positive, sometimes between populations it switched and could be negative. So we can see changes, evolutionary changes in how we respond to our partners. Okay, so this kind of interaction, so the swap from, or the switch from positive to negative, the fact that our 4 genotypes can behave differently, right? That we get very little shoaling with this partner and a lot with this partner, and it's the opposite when we change out the genotypes that are present, tell us that there's an interaction between the genotype and the social environment. And that's what I'm really interested in, and what I spent my sabbatical trying to understand. So I'm going to walk you through how we might get this kind of interaction. And rather than talk about indirect and direct genetic effects, which I know are pretty technical, instead I'm going to start talking about influence, so what a social partner is kind of putting out, and then responsiveness, how our focal fish might change her behavior. And so here's how it works. So if a fish varies for her influence, then we've got our kind of dull fish down at the bottom, and she causes a small change in her social partner's behavior. We might have a really charismatic, a super influential fish, and she causes a big change in her social partner's behavior. We might also have differences in how our fish respond to what their partners do. So the change that you just saw happen whereas a more responsive fish, given the same signal, might have a much more pronounced response. And so my lab has been focused on trying to understand what the physiological and genetic mechanisms are that cause a fish to be really influential and or cause a fish to be really responsive. And I'm just going to tell you that this is an important problem in evolution and in kind of biology more generally, because how traits are organized in the genome affects how they'll evolve. And right now I'm talking about behavioral traits in guppies, but these rules apply to traits that might affect human health or that have other phenotypic consequences. So it's important to understand kind of how the organization of the genome determines what happens. And what we know is that if there are lots of positives, we can get really rapid, big changes in evolution. And if there are lots of negatives in the ways in which these traits occur, then we get constrained evolution. Okay, so unfortunately though, we know almost nothing about how these traits are organized, which is called genetic architecture. So there's really just a couple studies. There's one in these little fast plants, which are called Arabidopsis, genus Arabidopsis, and there's a couple in literal Purdue chickens. And in both of these systems, they found a few genes that caused both an individual to be influential and other individuals to be responsive. So there's some evidence that it could be a set of genes that can do both things. So we've been, as I said, trying to understand what physiological traits underlie responsiveness and influence. And there are a bunch of pretty common candidates, so hormones, how much genes are turned on or off, so gene expression, their sensory abilities that might be related to things like, this is a picture of the fish's lateral line, so each of these little yellow dots are a pressure sensing bundle of cells. And I have this on here because my lab does a ton of work with this lateral line, but I'm actually not going to talk about it, but I wanted to show off one of these cool pictures that my students took. And so instead I'm going to focus on hormones because that's the most complete story that I have to date. Okay, so fish are pretty interesting in that they broadcast their physiological state continuously, and they do that by excreting steroid hormones. And steroid hormones are a pretty great candidate for creating both influence and responsiveness because how we respond to the environment depends on our internal hormone state. And then they're broadcasting these out and their partners can probably smell them. And so my lab spent some time investigating estrogen, the fish version of testosterone, 11-ketotestosterone, and cortisol, which is sometimes referred to as a stress hormone. And we found that there wasn't much evidence for 11-ketotestosterone or cortisol working in these cooperative behaviors, so we narrowed our interest to estrogen. And estrogen seems like it could be really important, not just because of our preliminary work, but also because it's known across vertebrates to modulate social learning and recognition, social motivation. It also regulates the synthesis of a peptide called isotosin, which is related to oxytocin, which is well known for causing pair bonding in mammals. So it affects pair bonding, affiliation and attachment, and again, social motivation and responsiveness. So we've got a lot of background that says that estrogen could be really important. So we set about verifying whether estrogen functioned in this system in a range of ways. Okay, so from now on, I want to emphasize that over the course of my time here at Stonehill, I've had more than 120 research students, and so I'm always going to now have pictures of the students who were the primary drivers of any particular experiment up in the corner, and whenever there's a black box around them, they wrote their senior honors thesis on data that are associated with the slide. And so these data were from Amy Hanlon and Anna Alcantara and a range of other students who helped them. And so they established that that signal that gets sent out to the environment varies both between inbred strains. So this is the total amount of estradiol that was excreted out into the water, which is also representative of the estradiol that was left inside the fish, right? So it varies both between inbred strains, and it varies between high and low predation populations. And in both of these cases, the strain and the populations that were the most responsive to their social partners, that were the most flexible in their behavior, excreted more estradiol. Once estradiol is either in the body or is taken in, and it interacts with a receptor called ER, so estrogen receptor. And Corey Mayer and Jackson Riley, and actually another student whose picture should be on here, Jim Cheney, verified that, again, there's a ton of variation in estrogen receptor expression, so how many estrogen receptors the fish are making. And that is true for wild strains, and it's true for inbred strains as well. And so how they receive and work with that estrogen is going to vary among individuals and among strains. So Roberto Rosa, who's up in the top, wanted to know whether we could sort of disrupt this kind of communication. And so he did a really clever experiment. So he actually used two different what are called endocrine disrupting compounds. So vanclozolin is a fungicide that's used on strawberries and grapes, and it acts against androgen receptor. And then he used BPA, which is pretty famous for coming from plastics, and it mimics estrogen, and so it makes the body think that there's more estrogen than there really is. And he used these two compounds to manipulate the physiology of our social partners. So he changed their phenotype, and then he measured whether the behavior of our focal fish, who was not exposed to anything, changed. Okay. And he had a ton of data. It was a big, complicated experiment. And I'm just going to tell you that this looks a little bit like confetti blew up, so, okay. The important thing to notice is that, once again, we took the behavior of the partners and we graphed the behavior of the focal fish in relationship to those partners. And then we've got the different lines. So these slopes are those coefficient of the interaction. And in the two cases, so these solid lines, those are the control fish, and then the dotted lines and dashed lines are the fish that were, the partners that were exposed to these endocrine disruptors. And what matters is that we are shifting how our focal fish respond based on what their social partners were actually treated with. And we can even reverse what they do by changing their partner's behavior. Okay, so what that means is that We have evidence for lots of variation here in our partners in a trait that can influence what our focal fish do. So we've got kind of both of these checked off. So then we got an upgrade in our technology based on both some funding from the National Science Foundation and also from a very generous gift from the Ferrara family. They are the family of an alum. And so we were able to get this auto tracking software. And so this is an example of our new data collection methods. And so what you're going to see is, again, we've got our predator model here. There's two little guppies, which are pretty hard for you to see, but what you'll be able to see are the trails that the computer puts in as it tracks them. And so they're swimming together. I've got this sped up. I mean, they are fast, but not usually this fast. They did an inspection, they cruised past, and then they went off. And Beth Parkinson, who graduated last year, so she did this with me as part of my sabbatical work, found that how well coordinated the partners were. So here we've got the distance between the partners. The shorter the distance, the closer they're swimming together, the more coordinated they are, dependent on an interaction between their two estradiol levels for females, but not for males. So the combined physiology of the two fish is what predicts how well coordinated they are. And so what we found, I'm just going to put these up so I can talk about them, was that if one guppy had high estrogen level and one guppy had low estrogen level, they were highly coordinated. If both guppies had moderate estrogen levels, they were moderately coordinated. And if both were high or both were low, they were poorly coordinated. And we think that what is happening is that estrogen is associated with flexibility. And so if you have one fish that's just going to do what it does, and the other one is flexible, then it can come to meet it. But if you've got two fish that are like super flexible, they're kind of chasing each other around, and it takes a long time for them to kind of coalesce to to kind of the middle. And then the moderate ones are, again, kind of in the middle. So they get to that coordination a bit faster. The males don't pay any attention. Okay. And this actually matched with, again, work that Jackson Riley and Jim Cheney did, where they found similar patterns in the male and female estrogen receptor expression. So that same kind of overshoot happens if you've got a whole bunch of estradiol and a whole bunch of estrogen receptor, then you're super flexible and you can often overshoot your partners. Okay, so what that means is that we have variation in estradiol physiology and variation in terms of the signal that they're sending out. It also means that we have variation in what they do with that information. And so what is really happening is that there's a two-way interaction going on here, right? Okay, but here's the thing. Somewhere between 15 and 25% of the vertebrate genome is coordinated by estrogen. So it's not surprising that estrogen matters, but what we don't know is much about what's happening sort of downstream, right? So we know that estrogen is changing, it's causing effects, but like how? So it turns out that a system that is really strongly modulated by estrogen, including in everybody in this room, is the visual system, because estrogen receptor expression affects how much opsins you are producing in your eyes, and opsins change what you can see, okay? And it's actually a really flexible system. So it turns out that visual sensitivity is able to change on the order of a couple of days in these little fish when they come into breeding season, and they need to make sure that that male is actually as like colorful and pretty as they think he is. It turns out that it changes sensitivity to hearing in songbirds, including through hair cells, which are what's in that lateral line that I showed you a little bit earlier. So we know that there are a bunch of systems and traits that are regulated by ER. This is just data from a different lab. This is Caitlin Friesen's work. And they just showed that using, again, kind of endocrine modulators, that they could change the expression of these opsins in related fish, closely related fish. And in fact, when we use those same treatments, we can change the visual acuity of the guppies. And so this assay was developed by Megan Machevsky and then was used by a range of students. And basically what we do is give fish an eye test. We let them swim in a rotating set of bars, and they follow them like they are partners. And when they lose sight of them because they can't clearly distinguish who they are, they stop following. And so if we modulate estrogen receptor, we can change how well they're able to follow their social partners. Okay, so the take home is that we already knew that cooperation evolves with familiarity and reciprocity. And that means that guppies are in these long-term, close associations with their social partners. That estrogen signaling and its effects on visual acuity influence cooperative anti-predator behavior, and that estrogen signaling could be both a direct genetic effect, responsiveness, and an indirect genetic effect, influence. And so it provides a really exciting potential mechanism for getting these kinds of interactions and for really understanding how cooperation might evolve. So we have a working hypothesis. So we think that partner visual cues combined with estradiol excretion is going to interact with the estrogen receptor expression, which affects opsin expression, which affects visual acuity, which then affects these cooperative behaviors, right? So we have this pathway to get from our social partners to our interacting phenotype in our focal fish. And the update is that, right, we'll put back in our lateral line signaling, and we're still in the process. This is a massive experiment. So what I primarily did on my sabbatical was to take 48 fish that came from 3 different populations, a domestic fish and 2 wild populations. I collected a hormone sample from them. I tested them with 2 different inbred social partners so that I could record how much their behavior changes. And then I sacrificed them and I collected the brain, where responsiveness should occur, eyes, gills, and body tissue. And we're in the process of doing what's called RNA-seq. So we're going to get a profile of all of the genes that are being expressed in every one of those tissues, in every one of those individuals. And we can use that to understand what is changing along with estrogen receptor, so sort of downstream from estrogen receptor. And hopefully I'll have an update on that soon. If I don't, just end up under my desk rocking because of the sheer amount of work that this requires. Okay. Okay, and then because I do a ton of work with the National Science Foundation, and the National Science Foundation always wants to know what your broader impacts are. I'm going to share just one of my broader impacts. And so I am particularly interested in increasing access to science education and careers in STEM for students and colleagues, particularly if they come from a group that has been historically excluded from science, and even more particularly from evolutionary biology. And I do that through creating substantive long-term research experiences and inclusive teaching and faculty development practices. And I'm just going to tell you about the first. So these are data for Stonehill. We have about 2,500 undergrads. On average, over the last probably 10 years, we've had approximately 18% students of color, 21% first-generation students. Women comprise 54%. I don't have data on LGBTQ students or disabled students. We have an overall graduation rate of 73%, and among the students who come into Stonehill planning to major in a STEM discipline, between 45 and 58% of them leave with a STEM degree. So more than this graduate, but they leave with a STEM degree. So sometimes they leave the science disciplines. And so I've had several kinds of National Science Foundation funding. So I had a career award and was part of a team that had an STEM, which is a scholarship award for students. And since I've been here, I've had more than 120 undergraduate research associates. I've had 41 seniors write theses. Students spend an average of 2.75 semesters and 1.47 summers in my lab, and then of course they then get to participate in manuscripts and theses and national meetings. The three over there, Hannah actually just graduated a couple days ago with her master's in medical genomics. Apsara is in a master's program for biotechnology, and Erica is working, she's in the middle, is working in biotech and planning to go into bioengineering, and that's a picture of them in Trinidad with me. And so the outcome of these interactions means that I have between 12 and 15 students in the lab at any given time. 30% of those students are students of color, 43% first gen, 4% disabled students, 59% women and non-binary students, 8% are self-disclosed as belonging to the LGBT STQIA plus community, and so a total of 77% come from groups that have been historically excluded from science. And within my lab, we have a 100% graduation rate and a 98% STEM degree completion. And so with that, there's obviously lots of people that I could thank, but I just want to point out that the undergraduates who specifically had data in this talk are the ones who are listed here. Here's some examples of their amazing faces. And then I have lots of collaborators, including people who are in this room. And then again, my funding came from the National Science Foundation, Stonehill College SURE, the Stonehill Father Francis Hurley CSC Endowed Chair, which I currently hold, and then gifts from the Michael, Ferrara, and Cassis families, as well as the Greater Boston Guppy Club. So with that, I'd like to thank everybody for making it all the way through this talk, and I would be happy to answer questions. Yeah, so thanks for the guppies. And then just to throw this out there, that is a picture of flamingos that I saw in the Caroni Swamp in Trinidad. One of my students very famously said that there's no internet, and Bleakley's idea of fun is going to the swamp. And I want to emphasize that that is the swamp. Okay, so. Brittany.
Okay, so I was interested in thinking about your wild type guppies versus the inbred strains and hormones.
Yeah.
So you have inbred strains that are bred in tanks, right?
Yep.
Versus wild types that
Yep.
are in streams with
Yep.
water flowing. So do you think that affects their sensitivity over time to these hormones? And could you in theory, like inbreed strains in like gigantic tanks or small tanks to see what that variation would be?
Yeah. So I think, I mean, their sensitivity is probably plastic, right? Because receptor expression is plastic. And so one of the things that I would like to investigate, researcher, Ryan Early, who works a ton on just fish hormones in general, like threw out an idea, and I have to like talk to him more about it. But one of the things that happens with the hormones is that, so I talked about just estradiol, so it's like unmetabolized hormone, but they make these metabolites that do other kinds of things, and one of them in particular is super persistent in the environment and super smelly. And so it's probably accessible to the guppies, even if they're in a river, because the females in particular like to hang out at the edges wherever there's like less water flow. I mean, they don't want to have to swim against the current all the time, right? So there's probably like pools. And then because there's a dry and a wet season, they also get isolated into smaller pools that can dry out. Yeah, so it's probably plastic, but we could select on sensitivity in probably all kinds of ways. Yeah, no, it's a great question. So plasticity means the flexibility in your trait expression. So something like height is not at all plastic, right? Like you get to your height and then you are, or there's maybe plasticity in your during your development, right? So like if you got fed enough, you'd hit kind of your maximum height. If you didn't get fed enough, you might not quite make your height. Whereas sensitivity might be able to be kind of dialed up or dialed down depending on the environment. Does that make sense? Yeah. Yeah.
I don't know much about guppies in general. The guppies here, or where do guppies live?
So they're native to Trinidad and that kind of northeastern edge of South America, so Venezuela and Guyana. However, they are actually invasive and feral in lots of different places because people have been keeping them in aquariums forever, and wherever it's warm enough, they can establish a population. And what you can't tell from those cute little faces is that they actually are voracious little predators. And so if they can put their mouth on it, they will eat it. And so, in fact, there are what are called feral populations in Australia where they keep trying to get rid of them because they eat the juveniles of native fishes. There are feral populations in Japan. There's one in a geothermal pool in Canada because it doesn't ever ice over. So they're tropical and they can live anywhere where it doesn't get too cold. And so they actually now are found around the world, but a lot of them are escaped, right? People dump their tank, and so they originate from an aquarium strain, a domestic strain, and then they expand into the native waterways. So there are lots of places, but I work with wild ones from Trinidad, which is where they're from and where they should be, and then domestic strains. Yeah. You would never, I mean, they really are like voracious little predators. Yeah. Yeah. So I think estrogen is the one that's really important. Cortisol and 11-ketotestosterone undoubtedly do lots of things, but don't seem to be causing a ton of variation in the cooperative behavior. But I am really interested in brain chemistry. So these aren't hormones per se, but the fish version of oxytocin is a really important target that we'll probably be working on next. So it's a great question. Yeah. They're very sad. They're never alone, right? And so they just, they do what I would characterize as stereotypic behaviors. So they literally just like rock. Yeah. Yeah. Yeah, it is. It's like lockdown. Like I really only isolate them if I absolutely have to, because it's just not good for them. And they die. Like they're so stressed that they die. Yeah. So I'll put females in a thing in order to get babies, and then like back in with the group. Yeah. Okay. Thank you, everybody. Hi, I'm Bronwyn Bleakley, and I am a professor of biology at Stonehill College. I'm an evolutionary biologist and behavioral ecologist by training, and I study a wide range of animal behavior. Today, I talked about work in my lab, particularly supported by my undergraduate students here at Stonehill, that has allowed me to explore how cooperative anti-predator behavior has evolved in Trinidadian guppies who work with social partners to determine just how dangerous big predatory threats are. And so we've tried to understand the physiology and the genetics that allows those behaviors to show up and to evolve. So I told my mom that I was going to be a biologist when I was 4. It's literally the only thing that I have ever really wanted to do. I did my undergraduate degree in ecology and evolution at the University of Arizona, and I studied evolution and behavior at Indiana University for my postdoc. So I guess I kind of knew that I would do something like this, but it wasn't until I was about halfway through graduate school when I discovered how cool guppies are, and I switched my research up, and then I've been kind of obsessed ever since. They're very accessible to the students as well, because we can do experiments that just involve putting fish in tanks and watching them swim around, all the way through, right, really complex, right, big data, gene expression, right, hormone analysis, right, micro dissections, and anatomical variation, right, and so the system is really kind of limitless, right? Like there are so many questions, we are never going to run out of stuff to do.